I don't know where the unmute button is. And took me at least like a minute to get it there. Well, thanks a lot for having me here. I guess, figuratively speaking, because obviously, we're freezing our bottoms here in Switzerland and you guys are having the good life over there in San Diego. So, it's clearly thank you for the organizers for having me here and thank you all for attending this talk. And I think it's very interesting that Gina and Vignesh and Simon didn't really take a no for an answer. And, when somebody said no, they just like, whatever, we're just going to do whatever the hell we think it's the right thing to do. And here we are now, right? So that's great. Thanks a lot also for being a bunch of rebels. Yeah, so I guess most of the times I tend to talk about protein design. Today, I will not talk about protein design. I will talk about small molecule design just because, every now and then one also has to kind of freshen up a little bit. And so that's kind of the idea. And the work that you're going to hear today comes from these wonderful five students that I have working with me here in Lausanne, which is Irini, Evgenia, Adrian, Ilya, and Arne. And I guess the title here is really about this conversation of how do we use different representations in order to interface the generation and how this can actually work all together, particularly in a molecular generation problem, which is a multi-objective optimization. So I guess, to design anything, any type of molecule, it's never just the one criteria. And so, and how do we balance this act in the best way we can? I think that's going to be a bit the common denominator of this talk. So as I told you, I will start just by telling you how we've been exploring different protein representations to understand protein ligand interactions and how we have applied this for fragment discovery. And then also, in this a second part where it's going to be about this multi-constraint problem for molecular generation. So yeah, I won't bother you too much with this part of the talk because I think that, every time that somebody sees my face, typically they think, well, here's this guy again talking about molecular surfaces. So yeah, molecular surfaces turn out to be super interesting. I don't know if they're very useful yet, or well, I think they're kind of useful, but but they're above all a whole lot of fun to work with, mostly because they seem to express a number of different properties on molecular structures, which are not absolutely trivial to express whenever we're just learning from atoms. Although, of course, this is a very debatable statement. But we've been working with multiple different types of representations based on surfaces. And here the idea is that, this surface is essentially the way that we create then these embeddings is that they learn about the shape and the chemistry of the molecules that we are analyzing. Now, one of the questions that we had, which we during the years we kind of like brushed off and never really worked on very seriously until a couple of years ago, was really, well, now since it's very hard to basically understand the patterns of molecular interactions, be it of protein-protein interactions or be it of small molecule protein interactions. And I would argue that the latter is actually much more subtle in a way than for instance, the protein-protein interaction problem. And it's extremely hard to actually have good tools that can tell us, well, molecule Y is going to bind to pocket X in in a specific protein. And that's most of what I'm going to show you today here. It's going to go around that problem. How do we figure out and which molecules bind where, and hopefully do that with an acceptable success rate so that these tools can in fact become useful. And the first thing that I want to tell you about today, something that we haven't quite published yet, but we've been talking around it for a couple of years now. It's what we call this fragment scope, and I would ask you to focus on this part of the slide, where the idea here was essentially to train these embeddings in order to understand which molecules or which kind of chemical matter is compatible with the surfaces themselves. And so the only task that this tool has to do is, given a protein pocket, I want to find fragments. And so for those of you that are not familiar with what fragments are, they're just typically small chemical moieties of very low molecular weight that typically are used in the process of drug discovery. So many times people, rather than starting already designing large small molecules, they basically start by assembling pieces of the puzzle and just putting fragments in the different pocket and then trying to link them so that they can reach high potency and high specificity. And here the idea is, well, how can we essentially try to have these embeddings trained and optimized so that we can distinguish very clearly the molecules that are the real binders from the molecules that are not the real binders. And so once we've trained these embeddings on the different surfaces or on the different subpatches of the pockets, we could eventually see that these embeddings have some level of chemical information of the ligands that they were supposed to bind to, and that they cluster broadly into the different classes, and that also in terms of discrimination between what are the real ligands that bind to specific patches versus that don't, that this is a test where we're having some success. Then once we do that, we try to ask the question, well, docking fragments because of their low molecular weight is actually a hard task. So can we actually now see how fragment scope in fact does this? And what we can see here is that compared to other approaches, it at least does as well as other approaches, which again, please also remember that this particular comparison is a difficult one, mostly because many of these other algorithms, in fact, have seen every kind of structure that it exists in the PDB, while we're just showing different fragments to the network. And essentially what you see here is that at least the performance of this fragment docking type of algorithm is at least as good as all the others, and in cases probably a slight better. What it also has that we were able to see is that we could actually generalize over the structures that we were giving as input. So we would not only find fragments that came from the same structure of or of the same protein, but we would also find fragments that came from different proteins that were completely unrelated, showing some kind of generalizability between the embeddings that were being learned. And so here I'm showing you this data for what you see on the x-axis. It's actually just the name of five model proteins which we've used for these types of benchmarks. Now, what's more exciting for us is actually to go to the lab and just try these things. And so I'm going to show you a couple of examples on how we can test this. So I will preface that these are actually extremely hard experiments to do, because in many situations, the fragments, when we're testing binding affinities of the fragments to the proteins, the affinities of these fragments are extremely low. And so because they're low, you actually have to do these experiments at very high concentrations of the small molecules. And what happens there is that often you can actually start getting non-specific binding when you're, for instance, throwing at a particular protein 500 micromolar or even higher concentrations than of the ligand. But so in this case, what you see, so we used KRAS as an example, one of these proteins that for now has a number of known ligands, but during many years has been considered undruggable. And here we've tried a number of different fragments, and what you can see is that whenever there's a blue box, it means that this fragment had a signal measured experimentally in a surface plasmon resonance type of assay. So it's not super important what the assay is, but you can actually kind of see that, well, is there a signal of binding here? And then you can test this at different concentrations. So typically more potent fragments would bind at lower concentrations. But then the question that we also ask is, well, so we are having a binding signal, but is this binding signal specific just to KRAS, or is also able to bind to other proteins, because we're testing this at so high concentrations that you never exactly know what's going to happen. And here what you see is that, in fact, there are other proteins which do not bind to this fragment. So at least there is some level of potency and some level of specificity that is given to this. Here you can also see that we have some fragments which are more distinct from the fragments that are known, and in this case is not extremely obvious, but what we're showing here is the Tanimoto distribution of these fragments, so how distant they are from other fragments that are known to bind to these proteins, because essentially we want to innovate in the chemical space. In this case, we actually maintain this relatively conservative. Now, in a second case, we also try this to PGK1, and so you see here that the profiles look relatively different. So we have a lot of hits at very high concentrations, also many hits at lower concentrations, but what you also see is that some of these hits not only hit this particular protein, but they also hit another proteins, which again is just essentially a measure of the lack of specificity of some of these fragments, at least at these particular types of concentrations. And here the Tanimoto distributions were a little lower, so showing that we are actually sampling fragments that are further away from those that were known for this target. Okay, and then of course the next step is, well, can we actually now link these fragments? And for this we use this tool that we published a couple of years ago, which was DiffLinker. Just a very brief explanation of what this how we actually framed this problem. But ultimately the idea is that we had a network that was able to figure out which linker size to build, and then it was able to connect the two fragments in order to ultimately create chemical matter between these two fragments, so that we could have a full-blown ligand. And so you can see here how this actually happens. And so just very briefly, we try this against a different target, which was now in this case BRD4. And what you see on the left are actually now crystal structures of ligands that we have designed, and you see the model versus the crystal structure. So you see that they're not exactly the same, but in many instances they are actually close in terms of the configuration that was predicted by the model versus the ligands that we actually were able to crystallize. So in this, we're having some type of success in terms of bringing these fragments into ligands. Now, beyond ligand design, right? How can we actually do this from scratch, right? So now in a scenario where you just have a pocket as an input for drug design, and you want to optimize, well, or you want to generate molecules that follow a number of different optimization constraints. And the optimization constraints are not small. So one of them here, just to give you an idea on how this how we think about this, right? But the idea is that, well, you'll have to optimize the chemistry of the ligand. That has to be valid. The protein-ligand interaction also has to be valid. It has to respect the geometry of the pocket itself, which is again related to the protein-ligand interaction pocket. And then there's another aspect which we typically don't take too much in consideration, and the way that we deal with this is, okay, so we just generate a ton of things, and then we just filter in the end whatever can be made chemically. And we really wanted to make an effort so that we could bring all these things together in an algorithm that would take all these constraints or restraints into consideration. And what Ilya, Arne, and Adrian actually came up with is what they call this large drug discovery model. Again, based on a flow matching architecture, where it basically diffuses both atom coordinates, but also bond types and bond lengths. And the conversation here is that they also have made a number of efforts in augmenting the data set. And ultimately what they ended up was with a model that could be repurposed for many different types of tasks in terms of small molecules, be it design and docking, fragment-based design, and constrained docking. So it can do a number of different things. And again, one of the problems that we face in generative tasks of small molecules is really that most of the molecules we do are just essentially not molecules that we can either purchase, or even if we would have all the chemists in the world, synthesize, because the chemistry just becomes too complicated. So one of the tasks that they in fact, or one of the ways that they in fact tried to optimize their algorithm was by using this class of techniques which are called as preference alignment. And here they really saw interesting results in terms of preference when they started trying the different ways of training this algorithm, particularly in the typical properties of QAD and synthetic accessibility. So just to give you an overview, that in principle, it seems like at least from the computational metrics that we can assess, the model seems to be doing what we want it to do. One thing that we think that is a problem that is far from being solved, again, also it depends who you ask, but if you would ask me, I would think that it is far from being solved. It's really this idea of how do we actually classify a small molecule as a binder versus non-binder in a very reliable way. So you're given a small molecule, you're giving its target, and now the task is, does it bind or does it not bind? And doing this with a very high success rate, I think it still remains an unsolved problem. However, here in this LDVM model, we're trying to quantify some of the uncertainty of the solutions that we have. I don't think we're completely there yet, but here you already start seeing that the idea is to go over these ligands and seeing by atom, is this a ligand that has like a high confidence or not. And what we see is that at least in terms of some of the physical characteristics, the score seems to express what it should express, since the bond lengths and bond angles need to be at a particular within particular ranges, and we see that the score does recognize that, as you've seen here in the latest plot. Now we then also started testing if this was good for docking. So here we also compared it with a number of different docking algorithms. So I won't spend too much time on this. Again, I also should just highlight that this is not global docking, but this is actually rather a local docking where you can place your ligands in pockets that you already know. And so here we're also performing relatively well in this task. You can see some examples here. But perhaps what is more interesting and exciting, and I don't know, I'm probably already running out of time, but I'm going to try to keep it pretty quick. The idea of how does this actually perform in terms of designing new molecules, and so Arne, Ili, and Adrian came up with a strategy for sampling here, which they called controlled sampling. And the idea is that every step that they actually add one new chemical group into the molecule that is being generated, they check if in fact this is a move that is going to take us towards a molecule that we can finally test in the lab when the trajectory is done. So that's the principle of this idea. So not super complicated, but the idea is that we're considering, what makes it very complicated is to consider all the different types of variations, which is, okay, what kind of interactions we're having. Is the strain energy of the ligand a good strain energy? Are the available building blocks there? So a number of different, so multi-parameter optimization really. But as we see here, oftentimes we end up with very interesting trajectories in terms of sampling. So when we sample, we can sample less, and maybe just one example here, we can sample less but obtain molecules which are aligned, let's say, with the properties that we're looking in terms of how much we need to generate the molecules that we're looking for, and ultimately we end up we end up with better solutions, or with more solutions that are actually synthetically accessible versus those that are not synthetically accessible and that we can actually purchase and test. And so speaking about testing, of course, I could not finish this without showing you some tests. So we started sort of like designing, again, now full-blown de novo ligands for some targets. So I'll show you first the KRAS. What you see here is some of the compounds that we've been testing in the lab from 1 to 5, and then you see a couple of positive controls. So do appreciate that the complexity of some of these ligands that are known to be high and potent inhibitors is not quite there present in our chemical matter that we're generating. And then here we evaluate the patterns of interactions between the known ligands and the ones that we're generating. And so you see that we, in fact, recover some of them. And so some experimental results is essentially when we tested 5 ligands, one of them was already 78 micromolar. of affinity, and we could actually map through a competition experiment, which I won't go too much into the detail, that was actually binding into the right spot. Now, for a different case, so the PGK1, which is also a very interesting case for us. Again, the same kind of conversation here. You get a few ligands that we tested in the lab, that we generated in the computer. These are the positive controls, and we can see that one of the ligands was already a 10 micromolar binder. Again, so these affinities are not affinities for which you would consider that these are drugs, but are at least good starting points that a medicinal chemist could look into and could start, well, okay, well, we have something to work with here, and hopefully we can quickly bring this to the nanomolar type of affinities very quickly. So here also experimental characterization according to NMR to make sure that the compounds are binding in the right pocket, and this is what we also observed here. Okay, with this, I guess just one thing that is still an important thing, although it's becoming easier and easier. So this generative AI, it still raises a lot of eyebrows in some of the users, and particularly with the people that are out there making molecules. And so in order to actually improve this aspect, we created a bit of a simpler interface so that people could actually go and input their pockets and very quickly and simplistically just start doing their own designs and creating their own types of chemical matter, which then they could go and iterate with the model over their own experiments to see if some of the suggestions that the model actually comes up with does make sense or do make sense, and try to basically then take them further into experimental characterization. And so here, just showing you more or less how this works, and how this can also be biased towards just changing different parts of the molecule rather than changing the full part. So a number of different features that one can have here. Okay, and so with this, a few summary points for you. So and maybe I will just show you that the two key points is that using the surface representations, we can have high success rates in fragment-based ligand discovery. And for the novel ligand generation, we very quickly generated several active ligands from very few tested candidates. This said, there's still sort of a number of important gaps that we need to fill, particularly the one from synthesizability is one that is very important. Okay, with this, my thank you for a number of different and specific collaborators that have been involved in this project through the years, particularly on the computational side. And also a big thank you to the group here in Lausanne, which has been working in this problem and a few others. So with this, I thank you for your attention, and I'll be happy to take any questions you might have.
Hi, very interesting talk. So I have two questions. The first question is about the process of encoding the protein surface. So as we all know, the protein ligand binding in this process is not static; it's a dynamic process. So if only encoding the surface representations, what if the information inside the protein is lost during this process, like the allosteric or some connectivity information inside? True.
Yes. So it's so I guess I interrupt you in your first point. It's one of the main limitations of what we do. It's really this picture or this static representation of the protein. So I think the reality of the fact is that molecules are not static, as you're going to hear from Anna, that molecules do move. The static representation is really just an approximation of the system to help us deal with what we don't know. That's basically the idea, because in all the reality, whenever we're talking about protein flexibility, we do not have good ways of actually knowing what the molecular motions of a protein are, because they're actually even experimentally extremely hard to measure. This said, I don't want to tell you that I'm going to run to the question, because I will try not to. At this point, we take a static representation. We also have some future efforts to at least encode or try to simulate or use other simulators to basically encode pocket flexibility and then extract from there perhaps a joint representation of an ensemble of structures. And that's how we're thinking to deal with this in the future. The other conversation to this is that sometimes these surface representations are in a way high information content but low resolution enough that they are permissive to different types of or different deviations of the pockets themselves, if that makes sense. And so sometimes we also see that even if the exact configuration is not there at the surface level, that matters a little bit less. That makes
Yeah.
sense.
So I guess my question is like related to, is there any benchmarking have done like between the surface representation and the complete protein structure representations?
Well, so I guess, there's probably many benchmarks that could be done. I can tell you one benchmark that we have done, which basically speaks to the problem that you're pointing, right? So at some point, we have tested if we have bound and unbound structures, do we see a degradation in the performance? And this, for this case, was actually for protein-protein interactions. It wasn't really for protein-ligand interactions. And there was in fact a degradation on the performance if we would not have the bound structure being represented as the target, if that makes sense.
Thank you so much for the talk. I have a question on the linker design using the diffusion model part. So how this fragment-based drug discovery is commonly done alternatively or traditionally is that you found two fragments and then you have generally have like a large library of linker design, and then you just like exhaustively sort of, perform the reaction in silico, right? So when you look at the results that comes out of that and then compare the results that might have come from these, the methods that practitioners usually use, do you see a good and similar alignment, or do you see a lot of the novelties in that? That's my first question.
Yeah.
And then the second question is that this constrained generative molecule design is incredibly exciting, but I want to understand about what are the things that could be reliably done today versus what is on the horizon now. If you can share your
Very good.
perspective on that,
Very
that would
good.
be great.
Yes, I will try. Again, I will preface it just by saying that it's my opinion. I think different people will have different opinions. But so let's go to the linker conversation, right? So in fact, we have started this without having a notion exactly of what the practitioners would do, and really what we started by doing was just to compare our approach to other approaches that were available. And one of the things that really came out, so there's two aspects. Oftentimes it was hard to get, even in the linker design problem, it was hard to get synthesizable molecules. That was one. And then in other, in a different aspect of this linker design problem, it was very difficult to have rigid linkages, if that makes sense. So oftentimes, you would end up with these linkers very long with rotatable bonds, and like this is really not what a typical high affinity molecule looks like, right? Because if there's a lot of entropic cost on the molecule, then it's difficult to have a high affinity interaction. So those were the two things where we saw diff linker making some contribution towards a linker design approach that was making more sense. That in fact, these molecules were viable compared to many other models that were creating these linkers, and if you would actually want to have more rigid linkages, you could actually do that. So those are two aspects. For the constraint generation and this type of multi design objective. So I will tell you my perspective, and I will be like fully straight to the point. I still think that the main limitation that we have currently is a scoring problem, which is I think generating things is simple. Generating good things is not simple, but generating things has become sort of like a bit standard of the field. Identifying which ones are really good molecules and which ones are not really good molecules is extremely hard. And also, of course, this idea of like good molecules versus non-good molecules. Let's just say binders. I'm not even talking about drugs. I think drugs is a whole other conversation that, these molecules have to have a number of features for manufacturing, for pharmacokinetics, pharmacodynamics. But I'm just saying, like, if you are given a set of molecules and you're trying to identify which ones are true binders and which ones are not binders, this is still extremely hard. So I think that would be my number one problem to work on.
Yeah, so because of the limited time, we'll move on to our next speakers. Thanks a lot, Bruno, for the great talk and the insightful answers.
Thank you.
Our first contributor